Good afternoon to all the EAPCET aspirants. This is your Prasanna Ma'am from NEET World, and today we are in our last session of the chapter Histology and Anatomy of Flowering Plants, and we are left over with one major concept, that is secondary growth. Okay, so yes, secondary growth is a major concept wherein frequently you got the questions in your EAPCET examination. So how you can exercise this concept, after which we will be exercising the complete EAPCET previous year questions. So here you look into first understand what is the definition of the secondary growth, what is secondary growth actually means, and how is it conducting and where is it conducting in the plant's anatomy, like in cortex region and in the stellar region. After which you have to know about spring wood and autumn wood, sapwood and heartwood, and the concept about complementary cells and lenticels. With this, and how a cambial ring formed in the dicot root and dicot stem. These are the concepts to be highlighted in the secondary growth. So now you see what is secondary growth in detail. Okay, what is secondary growth? Secondary growth is nothing but increase in thickness or girth of the plant by the activity of lateral meristems or secondary meristems. Take down beta, increase or take down, increase in the girth, increase in girth or thickness of plant by the activity of secondary meristems, by the activity of secondary meristems or lateral meristems. Fine, right? So this is what is it meant by what? Secondary growth, increase in girth or thickness by the activity of secondary meristems or lateral meristems. What are these secondary meristems and lateral meristems? Secondary and lateral meristems are those meristematic tissues which will appear in the later part of plant life to increase the girth and thickness of the plant. What are these secondary meristems? In the cortex region you will come across cork cambium, and in the stellar region you will come across vascular cambium. Vascular cambium. This vascular cambium can be interfascicular vascular cambium, interfascicular vascular cambium, or intrafascicular vascular cambium or fascicular vascular cambium. Fine, right ma. So this is about secondary meristems and lateral meristems. Now look into the types of secondary growth. Fine, right? Look into the types of secondary growth. Types of secondary growth includes stellar secondary growth and extrastellar secondary growth. Stellar secondary growth and extrastellar secondary growth. Stellar secondary growth occurs in the stele region in between vascular bundles. Okay, this occurs in the stele region in between vascular bundles. In between vascular bundles, okay, by the activity of vascular cambium. Fine, right? Now extrastellar secondary growth. Extrastellar secondary growth occurs in the cortex region by cork cambium. Cork cambium is also addressed as phellogen. Fine, right ma. And one more thing, in which type of plants or in which division do you observe the secondary growth. Secondary growth is not present in all the spermatophytes. Listen the statements properly because you can get a statement based question. Secondary growth does not occur in all the spermatophytes. Okay, that is all seed bearing plants. Okay, secondary growth occurs in the gymnosperms. Secondary growth occurs in the dicotyledonous class of angiosperms. Remember this statement carefully. Secondary growth occurs in gymnosperms and dicotyledonous class of angiosperms. Fine, right? So, secondary growth occurs in gymnosperms and dicotyledonous class of angiosperm. So, look into the definition of secondary growth important, types of secondary growth and what type of secondary or lateral meristems are involved in the growth and where do they occur. This is the basic information that you have to know from the secondary growth to address any kind of statement based question. After which you have to look into the secondary growth which occurs in the dicot stem and dicot root, and then from epidermis to the center, that is pith, and from pith to center after secondary growth what will be the anatomy and flow chart we will look into. So, first look into the dicot stem beta. Dicot stem secondary growth. So, here you see first epidermis region followed by cortex region, and then in the stela region you have primary phloem at the periphery followed by vascular cambium, that is intravesicular vascular cambium, and primary xylem, and at the center pith. This is the primary plant body transection, okay, transverse section of the primary plant body, that is primary stem. Now, after secondary growth what happens, you see the cortex region consists of what cambium? Cortex region will have cork cambium, okay, cork cambium. Cork cambium will undergo what activity? Meristematic activity, that is sequential meristematic divisions occur and fresh cells will be formed towards the epidermis and towards the endodermis, okay. Cells will be formed towards the epidermis and towards the endodermis. Those new cells which are formed towards the epidermis are known as cork cells or phellum. These are known as cork cells or phellum. And those cells which are formed towards endodermis are known as secondary cortex or phelloderm. Secondary cortex or phelloderm. And here you can see cork cambium is also known as phellogen. Fine, right, ma? So, after secondary growth you can see three layers in the cortex region. One is cork cells or cork or phellum, followed by cork cambium which is also known as phellogen, and this is followed by what? Secondary cortex or phelloderm. Three layers together we call them as what? Periderm. Three layers put together they are known as what? Periderm. So, periderm includes what, ma? Periderm includes phellum, phellogen and phelloderm. Okay, phellum, phellogen and phelloderm. Now, we are clear with the secondary growth that happens in the cortex region. So, the outermost layer here you can see it is phellum. Fine, right? So, the outermost layer over here is what, ma? Phellum is the outermost layer, followed by phellogen, that is secondary cortex, which is followed by cork cambium, phellum, cork cambium and phellum, phellogen and phelloderm. This is phelloderm. Fine, right. Now you look into the stellar secondary growth. This complete region is what? Stellar region. In stellar region, can you see primary xylem and primary phloem? Always. Here vascular cambium is present, right? This vascular cambium will undergo secondary growth. Okay, vascular cambium will undergo secondary growth and they will form new cells again. The cells which are as the result of meristematic activity will be formed. Those cells which are formed towards pith are known as what? Secondary phloem, and those cells which are formed towards the periphery are called as secondary phloem. Those cells towards pith region are secondary xylem and those cells towards the periphery are secondary phloem. So, here if you consider the stellar region, here you see ma, in stellar region what happens? First outer side it is pericycle and inner vascular bundles will be there, right? See, vascular bundles. Yeah, here you see in between xylem and phloem, primary xylem will be towards the pith first. Yes or no? Primary xylem which has interfascicular cambium is present, and towards the peripheral end phloem is present, right? Now this meristematic layer intrafascicular cambium, here the cells which are present in between, and from pith we know pith rays will be formed. Those are medullary rays, right? So adjoining to the medullary rays and adjoining to intrafascicular cambium, interfascicular cambium is built like this. Okay, interfascicular cambium is built like this, adjoining to intrafascicular cambium. So here the interfascicular cambium, and intrafascicular cambium, okay, both of them together they will form what? Cambial ring. This cambial ring will undergo what activity? Meristematic activity, and it forms cells towards the pith region, that is above the primary xylem towards the pith region. In close proximity this region is having what? Secondary xylem. Fine, right? And above the vascular cambium, below the primary phloem, what cells are formed beta? Secondary phloem cells are formed. Secondary phloem cells are formed. That is what is given in your diagram. So from the pith after secondary growth, if you observe from pith region at the center, what will be the first layer from pith? That is primary xylem. Primary xylem followed by secondary xylem followed by a vascular cambium, and then secondary phloem followed by primary phloem and then pericycle. This will be the stellar pattern of layers after the secondary growth. Now in a dicot stem you have understood extrastellar secondary growth and stellar secondary growth, right beta? So here we will see from epidermis to pith region what will be the flow of layers in a dicot stem after secondary growth. Here we go. First is what ma? Epidermis region. Yeah, first is epidermis region. First is epidermis. Epidermis followed by cork cells, cork or phellum. This flow chart is very important. You take down, if not take a screenshot of this. After cork or phellum you will have cork cambium or phellogen, cork cambium or phellogen, and then you see this is the cambium, this is the region where cambial activity is taking place, right? And this is followed by what? Secondary cortex. Secondary cortex or phallodome, and then endodermis, and then pericycle. Close to the pericycle, primary phloem will be there, because close to the cambium, close to the vascular cambium, you will have secondary phloem. So, here pericycle followed by primary phloem, followed by secondary phloem, followed by vascular cambium. Okay. So, here vascular cambium activity is present. This vascular cambium followed by secondary xylem, followed by primary xylem, and lastly at the center, that is pith. Clear, right? This is the complete flow chart that you have to remember for sure for dicot stem. You can take a screenshot of this beta, very important, very, very important. Clear, right? Done? Clearly understood the dicot stem secondary growth? If it is done, we will move on to dicot root. In dicot root also the scenario will be same in the cortex region, but then the cambial ring which is formed in the dicot root will be wavy cambium. Okay, in dicot root, wavy cambium is formed. Fine, right? So, this wavy cambium is formed in the dicot root. How? If you see the stellar growth, stellar secondary growth in the dicot root, stellar secondary growth in the dicot root, if you see, first one, this is the endodermis, single layer which has tuberant depositions in its radial and tangential walls, and after this if you see pericycle is the layer. Okay. From pericycle region only, here pericycle, vascular cambium will arise. So, in dicot root, from pericycle region, what all will arise? Vascular cambium, and lateral roots arises. Fine, right? So, from pericycle region, vascular cambium and lateral roots will arise. So, here from pericycle you can see the vascular cambium arised, and here what type of vascular bundles are there beta in dicot root? Radial vascular bundles are there, radial type of vascular bundle arrangement, wherein xylem and phloem are not at the same radius, but they are side by side to each other. So, the cells which are present or the tissue which is present in between xylem and phloem is known as conjunctive tissue. So, here the cells from the pericycle and from the conjunctive tissue will get meristematic, and a wavy cambium is formed like this in between xylem and phloem. Okay, wavy cambium is formed like this in between xylem and phloem. And again the chart will be same in the stellar secondary growth. If you consider from pith, close to the pith region, of course primary xylem will be there, and next to the primary xylem you will have secondary xylem. Next to the secondary xylem you will be having vascular cambium. After vascular cambium it is secondary phloem, followed by primary phloem, followed by pericycle, followed by endodermis, and then secondary cortex, and then cork cambium or phelloderm, cork cambium or phelloderm, and then it is cork or phellum. Then at the periphery, the ultimate final layer is epidermis. This is the flow chart from pith to the epidermis of dicot root. Take a screenshot of it, ma. I hope the wavy cambium concept is clear to all of you. Yes, right? Clear, right, ma? Yeah, now you see. Now dicot stem is done, dicot root is done, and now see in the tropical regions where there will be variations in the seasons, the secondary growth is not even, okay. Secondary growth is not even, it is dependent on the seasons. As such, the secondary growth which occurs in the spring season is known as springwood or earlywood, and the secondary growth which occurs in the winter season or autumn season, this is called as autumn wood or late wood, okay. As such, two types of woods are formed based on the season in which the secondary growth is occurring. As such, you see the clear differentiation between spring wood and autumn wood, because many a times we got questions from spring wood, autumn wood and heartwood, sapwood. As such, here you will get what? Layers. One complete ring of spring season and autumn season. This is known as one annual ring. Annual ring. Okay. This is spring wood, one spring wood and one autumn wood is known as one annual ring. Okay. Clear, right? So, as such, based on the number of annual rings formed, we can count the age of the plant, and that concept is known as dendrochronology. Okay. So, this concept you have to be clear with. One spring wood and one autumn wood will form one annual ring. Based on the number of annual rings formed, you can count the age of the plant, that is under dendrochronology. So, here you see spring wood or early wood and autumn wood and late wood. The differentiation should be very clear. Firstly, it is formed in the spring season. So, it has wider lumens and low density of the vascular bundles or vessels. Okay. And here, as it is in the spring season, you can see more number of xylary elements and the cambial activity is very active. Spring wood vascular cambial activity will be very active. It has more xylary elements, it has wider lumen and high water requirement is present. Okay. High water requirement is present over here, and then it is light colored, soft and porous wood texture will be there, and transition from spring wood to autumn wood, when the season gets changed, transition from spring wood to autumn wood is gradual, or you can say slow. Okay. And this forms inner light ring. Spring wood is what? Inner light ring. And if you see autumn wood, as it is in the winter season, the cambium will be less active and it has few xylary elements and narrow lumen vessels will be present and low water requirement with high density. It is dark colored, spring wood will be white colored. This is hard and compact, whereas spring wood is what? Soft and porous. And here transition from autumn wood to spring wood is sudden or fast. Okay. And then this forms outer dark layer, spring wood forms inner light layer. If you want, you can take a screenshot of this very important slide to see. Then you see ma, here sapwood, which is also addressed as alburnum, and heartwood, which is also known as duramen. Okay. Sapwood is nothing but the peripheral or outer region of secondary xylem is known as sapwood. Okay, and here age of the tissue is younger secondary xylem. First of all, you understand what is sapwood and what is heartwood. See, as a result of cambial activity in the spring season or autumn season, you will get the sequential layers. Okay, one circle of spring wood, one circle of autumn wood. As such, the secondary growth is built. Once the girth of the stem is increased, the central region will get dark, and that is called as heartwood. Okay, heartwood or duramen. And the peripheral region, which is soft and light, is addressed as what? Sapwood. This is peripheral outer region of secondary xylem, and this is inner region of secondary xylem. Heartwood is what? Inner region of secondary xylem. And age of the tissue, it is younger secondary xylem. Here older secondary xylem will be there. This is light colored, heartwood will be dark colored. And storage tissues, living parenchyma, here in the sapwood, this will conduct water and minerals. And in heartwood, dead cells will be there, they will not conduct any water or minerals. There is no conduction activity in the heartwood, whereas some tracheal plugs will be arised from xylem parenchyma. These are known as tracheal plugs or tyloses. They will store the food material. They will store food material. Okay, they will arise from xylem parenchyma. We can expect a question from tyloses or tracheal plugs. These are also known as what ma? Tyloses or tracheal plugs. Fine, right? And then you see deposition of substances. There is no deposition of substances like resins, tannins and gums, but then here in the heartwood or duramen, conduction will not be there, but storage of food materials in the form of some secondary metabolites like tannins, resins, oils and aromatic substances will be there in the duramen. Mechanical strength is less hard in the sapwood, mechanical strength is hard and dense, strong in the heartwood. Okay, and here in the sapwood, resistance to pests, it has low resistance. Heartwood, as all these storage constituents are present, it has high resistance for the insect attack. And main function here is conduction. Here main function is mechanical support, storage and protection. Here this gradually conducts to heartwood, whereas heartwood is permanently non-functional. Heartwood will never convert to sapwood because sapwood is at the peripheral region. So long the secondary growth is increased, the central portions is converted to the heartwood. Heartwood will be never converted back to the sapwood. Okay. So clear with the information about sapwood and heartwood ma? Clear with this? You can just take a screenshot of this and analyze the differences properly. Very simple and very easy. Clear, right? Now you see the concept of lenticels. So you are clear with the secondary growth, where does it occur, how does it occur, by what meristematic tissues, how secondary growth occurs in the dicot stem, how secondary growth occurs in the dicot root, and then what is the differences between early wood and late wood, okay, that is spring wood and autumn wood, and then sapwood and heartwood. Till here it is clear, no ma? Now we will get to know the concept of complementary cells and lenticels. So here what happens is, because of the sequential secondary growth of cork cambium or phyllogen, okay, sequential secondary growth of cork cambium or phyllogen, what happens is fresh cells are formed towards the epidermis. Those are called as what? Cork cells or phellum, and those cells which are formed towards the endodermis is addressed as what? Secondary cortex. But because of the vigorous activity of cork cambium, the parenchymatous cells which are present in the cortex region will get ruptured. Okay, parenchymatous cells will be ruptured and the epidermal parenchymatous cells also will be ruptured because of the bulk length of cork or phellum. So those cells which rupture these spaces, they are known as what? Complementary cells, means additional cells which are formed, which are making the epidermal wall ruptured, and the spaces which are created in the epidermis, this is what? Lenticels. Okay, this is lenticels which will help in the exchange of gases. Exchange of gases. Thereby serve in the purpose of what? Breathing of plants. Clear, right? So here cork cambium and the cells which are towards the endodermis secondary cortex, those cells which are towards epidermis, these are complementary cells, cork cells, and additional cells which are formed because of the vigorous activity of cork cambium which rupture the epidermal parenchyma cells. Those cells are complementary cells, and here small spaces which are created, breaks or spaces that are created in the epidermis, they are known as what? Lenticels, which will help in the exchange of gases, that is for the breathing in the plants. So this is the complete concept of what? Secondary growth ma. Now we will move on to the EAPCET previous year questions. Okay, open to all. This session is open to all. EAPCET PYQs. Everybody can answer ma. Look at the first question. Yeah, here identify the monocot root characters. So we need to identify the monocot root characters. Polyarch condition is observed in monocot root. Yes, diarch to tetrarch in dicot root. Large pith is present in the monocot root. Pith is small or inconspicuous in dicot root. Present of bundle sheath cells. Bundle sheath cells are present in monocot leaf. Scattered vascular bundles are present where in the monocot stem. So here what are the characteristics which are associated to monocot root beta? 1 and 2, that is option B. Look into the next question. Very simple, asked in 2023. Conjoined, collateral and closed vascular bundles. Okay, conjoined, collateral and closed vascular bundles. Conjoined where xylem and phloem is in the same radius. Collateral where xylem and phloem are side by side to each other, and closed vascular bundles, they are present in where? Monocot stem. Okay, conjoined, collateral and closed vascular bundles are present in monocot stem and monocot leaf also. If it is given, it is monocot leaf and dicot leaf also we can choose. And then which of the following characters? Here I would like to address the vascular bundles of others also ma. Monocot root, it is radial and closed. Dicot root it is radial and open. Dicot stem, it is conjoined, collateral, open type of vascular bundles in dicot stem. Here you see third question. Which of the following characters are found in hydrilla stem? Hydrilla is what? A hydrophyte plant, right? Okay, aquatic plant or hydrophyte. Parenchyma is present which helps in buoyancy. Sunken stomata is not present because sunken stomata you can see in xerophytes. Palisade tissue, no. Xylem activity will be there. Okay, palisade tissue you can see it, and xylem activity is also present. So here you can choose 1 and 2 you cannot choose because sunken stomata is observed in xerophytes. Stem he is saying, no. So palisade tissue will be there in the leaf, not in the stem. Xylem cavity will be there in the stem. Fine, right? Because that is schizolysigenous in origin, cavities will be present. So it is 1 and 4, option B. This you see, fourth question. In which of the following plant materials polyarch condition of xylem can be observed in the transverse condition? Polyarch condition of xylem is observed in what? Monocot root. Very direct question. Option B. Next question ma? Yeah, look at this statement based questions. Which of the following tissues are present in the dicot stem but absent in the monocot stem? Present in dicot but absent in monocot. If you see in dicot stem, collenchymatous hypodermis is present, starch sheath is present in the endodermis, right ma? So yes, these are present in the dicot stem, absent in the monocot stem. Sclerenchymatous hypodermis is present in monocot stem. Large central plinth closed vascular bundles in monocot. Epidermis and conjoint vascular bundles, this you see in the monocot, okay. Monocot and dicot, in both you will see the epidermis and conjoint vascular bundles. He did not mention whether it is open type or closed type, but then conjoint you can observe both in the dicot and monocot stem, right ma. So here the option which is suitable only for dicot stem but not monocot stem is option 1. Then you see next question. Identify the incorrect statement among the following. Remember students, whenever statement based question is given, first you have to identify, first you have to analyze whether in the question they are asking you to identify incorrect statements or correct statements. Based on that you have to go for the procedure on solving the question. Just like that, peripherally do not see the question and go for the options. Observe carefully whether he is asking you to identify correct statements or incorrect statements, right ma? So here if you see in the 6th question, the question is asking us to identify incorrect statements. Look into the statements properly and then answer. Here we go. Epidermal cells are parenchymatous and have small amount of cytoplasm. Yes, no, parenchymatous cells will not have small amount of cytoplasm. Gymnosperms have vessels in their xylem. Vessels are absent in gymnosperms. Bicollateral vascular bundles. Phloem is present on the either side of xylem, not cortex. So this is wrong. Root endodermal cells have depositions of water impermeable suberinous Casparian stripes. So D is correct. You can eliminate the option C here. And then epidermal cells are parenchymatous only. Yes, parenchymatous cells will have large vacuole, but cytoplasm description is not given. So yes, you can consider this option as correct. And gymnosperms have vessels, so it is completely wrong. And then bicollateral vascular bundles have phloem in between xylem. Xylem is sandwiched at the center by 2 phloem tissues, that is bicollateral vascular bundles. There is no involvement of cortex over here. So it is B and C, option B. Okay. Next question ma, statement based again. Here we go, identify the correct statement. So here we need to identify the correct set of statements. Periderm consists of phellogen, phellum and phelloderm, true. Permanent tissue having many different types of cells. Yes, permanent tissue has many different types of cells is called as complex tissue, true. In stem, endodermis stores starch grains. Yes, in dicot stem it stores starch grains, okay. Heartwood and sapwood produced in a year have same color and density. No, they do not have same color and density. So here the right options are A, B and C. We will go with option A, okay. Next question. You need to identify A, B, C, D options based on the statements they have provided. Here we go. Large colorless empty cells along with leaf veins are used for absorption of water. These are what? Bulliform cells. Next, cells much elongated, unbranched, with pointed needle-like apices and thick cell wall, and are dead at maturity. So these cells are what? Fiber cells, because they have needle-like apices. You can directly go and dead at maturity. So phloem fibers. The cells which are longitudinally arranged with perforated end plates. Longitudinally arranged with perforated end plates will be what? Sieve tubes, which have what? Sieve plates at both the ends. Cells left behind from shoot apical meristem. What are the cells that are left behind the shoot apical meristem? In the axial region, those are the axillary bud. So here one is C, 2 B, 3 is D, and 4 A. So here 1C, 2B, 3D and 4A. Option C is the right answer. Clear, right, ma? Yeah. Next question. Multi-column based match the following type of question. Lignified walled dead cells. Okay, dead cells with lignified wall are seen in the heartwood region and they do not conduct water. Okay, so this is identified. Next, during unfavorable season, plant cells are less active with less number of xylem elements with narrow lumen. This is what? Autumn wood. Okay. Next, cambium forming narrow band of parenchyma. Narrow band of parenchyma, that is of pericycle to protoxylem. Okay, pericycle to protoxylem, and it is about medullary rays which conduct the radial conduction. Next, cambium forms below the phloem. Below the phloem, that is in between secondary xylem and secondary phloem region, right? So it is what? Secondary growth of the root. Fine. Below the phloem, it is secondary growth of the root. So here if you look into the options carefully about heartwood, these are the two connecting dots, and in the unfavorable season, plants will be less active, and that is about autumn wood. And cambium, that forms narrow band, it is pericycle above the protoxylem and medullary rays. Okay. Secondary medullary rays are also known as vascular rays. Okay. They are formed along with the vascular cambium, and they have secondary medullary rays as well because of the vascular cambial activity in the interfascicular region. Secondary medullary rays are formed whose main activity is the radial conduction. And then cambium forms phloem, yes, correct, between secondary xylem and phloem, and here secondary growth of the root. It is option A. Okay. And next, 14th question, correct statements among them. Correct statements we need to identify. Ring arrangement of vascular bundles is character of dicot stem. Yes, ring arrangement of vascular bundles is characteristic of dicot stem, correct. Yes, vascular ring, cambial ring is formed here. Next, the root hairs are branched and some are secretory. The storage cells in xylem are thin walled, made up of cellulose and stores tannin substances. Yes, it is about xylem parenchyma. Parenchyma cells generally present in medullary rays. No, parenchyma cells are not present in the medullary rays. So here we can go with option A, B and C, right? Yeah. Next question. Assertion, reason type of question, you can answer it. Assertion, reason type of question. Primary and secondary phloem arranged, crushed in the secondary growth of dicot stem. Yes, in dicot stem, primary and secondary phloem are crushed in the secondary growth. No, it is primary xylem and primary phloem are crushed in the secondary growth because they will be at the extreme ends. It is not primary and secondary phloem. It is both primary xylem and primary phloem. Okay, secondary xylem is produced more than the secondary phloem. This reason is true. So here A is false and R is true. Clear, right, ma? Next questions. We need to identify again. A cell thickened at the corners with pectin, hemicellulose, cellulose depositions are what? Colon-chymatous tissue, which is option B. Next, phloem is present on the either sides of xylem separated by cambium. Here he is talking about bicollateral, okay, vascular bundles. Okay, so 2 is C. Cells with waxy thick layer preventing water loss. Waxy thick layer preventing water loss, that is seen in the epidermis. Just a second. Yeah. That is cuticle, right? Epidermis cuticle, which is A. And then protoxylem towards the center, metaxylem towards the periphery of the organ. Such type is called as endarch, which is observed in the stems. Fourth is what? Endarch, which is seen in the stems. So here B, C, A and D, option A. Clear, right? Yeah. Next question. Here options are not provided, but then you can easily identify the correct match. Okay. Look at it carefully, ma. Springwood, where xylem vessels are wider, they have wider lumen, and heartwood you can see highly lignified secondary xylem. Starch sheath. Starch sheath is observed in the endodermis of dicot stem. Periderm includes what? Phellum, phelloderm and phellogen. Clear? Next question. Again match the following type only. You have to identify between these two lists. Highly lignified dead cells with narrow lumen. Dead cells with narrow lumen, you can see it in monocot stem. Highly thickened dead cells with narrow lumen. Look into the other also. Lysogenous cavity, this is 1, 2, 3 and 4. 2. Lysogenous cavity is observed in monocot stem, that is A. Radially placed parenchymatous cells between vascular bundles, these are medullary rays. So 3 is C. Polyarch xylem bundles are observed in root. So 4 is D. So 1 will be B, okay, that is leaves of tea, because they have what? Sclerites. They have sclerites. So here 1 B, first it is B, then A, then C and D. This will be the correct flow. It is option B. Next question, ma. Of course, here answer is already given. You look into this properly. If you are not getting, just let me know. Next. What is living mechanical tissue in plants? Living mechanical tissue in plants is parenchyma. Next, assertion and reason type of question, you see carefully. Estimation of the age of tree by counting the number of annual rings is called as dendrochronology, correct. Periderm is formed by association of phellogen, phelloderm and phellum, true. So both statements are true, but nowhere it is nowhere the reason is explaining the assertion. In this case, you will go with the option B. Next, conjoint, open and end arc type of vascular bundles. You will see these are common in where dicot stem. Next, assertion reason type of question. Secondary phloem is produced more than secondary xylem and this forms a compact mass in dicot stem. Wrong, because always it is secondary xylem that is produced more than the secondary phloem. The cambium is generally more active in the inner side than in the outer side. True, but here the assertion is wrong and reason is true. So, here we will go with the option D. Last slide of today's session, ma. Look at these questions, assertion reason and a basic statement based question. First, in the assertion, spermatophytes contain a type of primary xylem which is exarch in stems. Not in all spermatophytes, right? Spermatophytes stem he is saying. In stem it is not exarch condition, in stem it is endarch. So, here assertion is wrong. In reason you see, protoxylem towards periphery, metaxylem towards the center, such arrangement is known as exarch condition. True. So, here again A is false, R is true. Vascular cambium produces what, ma? Vascular cambium will produce secondary xylem and secondary phloem, option C. So, with this, complete chapter of histology and anatomy is done with the EAPCET previous year questions. This playlist will be highly helpful to all the students who are attempting EAPCET 2026 exam, where the complete content with the PYQ practice is provided for free in this public platform. So, use it, students, and then next we are going to start with the chapter modes of reproduction. So, stay tuned and thank you.